Yeah, thank you very much, organizers, for giving me opportunity to show my work. Can you guys hear me online? Okay. Okay, great. So, as my title says, I'll be talking about the tumor microenvironment, and during this week, a couple of people already mentioned the importance of tumor microenvironment in cancer and treatment and so on. So let me tell you what we are doing in my lab. Okay, it works. So why the tumor microenvironment matters? So historically, cancer has been viewed as the disease of cells, right? So whatever the unrestricted growth, the differentiation, that there was thought that everything is caused by gene mutations. Even the progression or invasiveness, it was thought that this is determined solely by the factors within the cells. But, with time, biologists also realized that all the external factors that surround the tumor can play the role. And I want to show you one example from Valerie Weaver lab from the Berkeley, UC Berkeley. When they were growing the cells, MCF10A cells, and the next slide I'll show you a little more about those cells, which are non-tumorogenic. And when you grow them in the 3D in vitro setting, they form spheroids, and then spheroids which are empty inside, so they have lumen, and they grow them like in the normal microenvironment. They form those nice spheres, but when the stiffness of the gel that they are growing in is increased, they start behaving like tumors. So first they could fill in the duct, there is no lumen anymore, and then if you increase the stiffness even more, then they start migrating, when normally they are epithelial, so they would not migrate, up to the point that they really look like the invasive tumors. Those are the same cells, there is no genetic changes, that microenvironment is different. So those MCF10As, this is the cell line. The MCF comes from the Michigan Cancer Foundation, so that was the non-tumorogenic epithelial cell line isolated by this foundation. And the schematic shows you that when those cells are put in the 3D gel, they first form the sphere, and on the bottom, there are images from our collaborators from Vanderbilt. So they first form the full sphere, then the cells on the outside differentiate, and then the cells in the middle are triggered by apoptosis, and they will die. The apoptosis was mentioned here, the programmed death. And then what you end up with is this shell of cells with the hollow lumen. And this is a movie again from our Vanderbilt collaborators. And I hope you will see that this is only a shell of cells outside and the lumen inside is hollow. And this cell line is meant to represent the normal development of the breast duct or the acini. It's non-tumorogenic means if the biologists put this into the mice, they will not grow the tumor. So with this non-tumorogenic cell line, if you put them in different microenvironments, they start behaving like invasive tumors. So that's what, our lab wants to focus on, the tumor microenvironment. And by the tumor microenvironment, we mean the area around the tumor or within the tumor in which the tumor cells will interact with the tumor microenvironment components. And we wrote this review a couple of years ago with Lisa Macaulay, who is a biologist. So we have to come to the consensus what we want to include as a components of tumor microenvironment. And we come up with those 5 classes. So there is a host tissue, and somebody already mentioned, tumors don't grow in the vacuum. So if we are modeling, and most of our modeling in my lab are in the context of tissue, so there is a host tissue structure. The primary tumor especially, they grow in the ducts, they can grow in the skin, there will be layers of cells, they grow in polyps. The second component is the cellular component. So all the cells, not tumor cells, that you could find in the microenvironment. And we come up with this picture, which you see is pretty busy, has many different cells, macrophages, lymphocytes, and then lymphocytes have many different categories. The endothelial cells that will form the vessels or the lymphatic system, fibroblasts, and so on, fat cells. The third component is what we call the physical component. So the structure of the extracellular matrix. So again, the cells are not in the vacuum. There is a lot of structure composed with fibers, and it's, of course, it's complex too. So there could be collagen, elastin, laminin, fibronectin, and many other proteins that form the ECM. The next one is the chemical structure, and there was a couple of talks already of those components. There could be oxygen or acidity, could be growth factors, cytokines, MMPs, matrix metalloproteinases, all the components that could diffuse in the extracellular matrix or could be bound to the extracellular matrix. And then finally, the fifth component is the fluid structure. So again, if we are talking about the diffusing, this diffusion is not in the vacuum. This is in the fluid that is interpenetrating the extracellular matrix. So what I wanted to tell you today is I briefly show you this micropharmacology framework that we are using for all those problems. And then there is 5 classes. It's a little too much to talk about each of them separately. So I thought I will focus on the 2 that are maybe less often used. So the stromal fluid structure and the stromal physical structure. I will show you one example. So for the fluid, something that we published a couple of years ago. For the ECM, something that we published last year. And then at the end, I will tell you about our ongoing work that will combine the host tissue architecture, the bladder, mice bladder and tumor, the chemical structure and tumor oxygenation, and then stromal structure with the cytotoxic lymphocytes. Okay, so the micropharmacology framework that we want to use, it's kind of depending on the problem, we can use different modules. So we are interested in the tissue architecture, so we will have the tissue structure. When it comes to the drug or metabolites, we could incorporate the discrete drug molecules or metabolites molecules or continuous drug concentrations. When it comes to the transport, we can include the advection with the interstitial fluid or diffusion or the combination of both of them. And then we want to have those molecules effect on the cells, so we could have the membrane diffusion uptake or receptor binding, or there may be no uptake at all. So in this, again, like we wrote this a couple of years ago, so we put the illustration with the cells which are circular, but now we have more complex situations, so we actually discretize cells from the histology, and we can also add the extracellular matrix structure in those models. Okay, so I will start with the fluid structure. Why fluid? So when I start, so my background is fluid dynamics. I was happy to do this part, but when I start actually looking into that, I was also surprised. About 20% of our body is fluid, and for example, in the skin, the fluid takes 50% of the skin. In muscles, is of course less. So this interstitial fluid consists of the water and then the solubles in the water. It's created by this transcapillary filtration. That means the vessels, especially in the tumors, are leaky. So there may be plasma leaking from the vessel into the tissue. And then the sink for those are the lymphatic vessels. So you have flow from the vessels from the blood vessels into the lymphatic vessels. And this is very slow flow, about 1 micrometer per second. And what experimental results have found, that this flow has effect on some cell differentiation or cell migration. So they found that the fibroblasts, when they are exposed to this shear flow, they could differentiate. They also found that the cells, the tumor cells, can secrete certain factors, and when those factors are moving with the fluid towards the lymphatic vessel, the cells are following this gradient and enter the lymphatic system easier and then metastasize. And of course, as I said, the fluid is needed for the transport of nutrients, waste products, cytokines, and so on. So there are different techniques to use in the fluid dynamics. What I will be showing you are the regularized Stockslets methods. So the motivation for this project was our collaboration with David Morse from Moffitt when they developed the fluorescent biomarkers. And for this project, we will look at the tumor structure. We will have the discrete molecules as our biomarkers. We will have diffusion and advection, and there will be receptor binding. So part of this whole framework. So we wanted to model tissue structure. So on purpose, which we look at the histology, we selected the region that the capillaries on the left that simplify our simulations. And that was published a couple of years ago, a long time ago. So that was our initial step. Then we will build this tissue, we will have influx of interstitial fluid. We want to calculate this flow in between the cells in the interstitial space. If you look at this ratio that is called the Reynolds numbers, there is a density and viscosity of water, and then NLU are the characteristic values for the length and velocity. So the length here is in microns, 10 microns, maybe 100 microns for the cell size and the domain size. The velocity characteristic value is for the fluid flow, so it's about 1 microns per second. So we are in the really low Reynolds numbers here. So what we want to use is the Stokes flow, which is governed by the pressure, differences in pressure, and then implied force. But if you apply force to the point, the solution to that has the singularities. So what we will be using is the regularized Stockslets method, which was developed by Ricardo Cortés, one of my professors in my PhD at Tulane University. So that means that instead of the point force, you spread this force into the small region, and you usually use this blob function that have like a Gaussian structure. And if this function, the spreading function or the blob function is chosen properly, then you actually can solve the equation exactly, right? So you don't need any approximations here. You have the exact solution that depends on the point forces that are applied in the whole problem. So this is even nicer because if you write this velocity in terms of forces, this is really linear equation. So what we can also do, if we know what velocities do we want, we can solve the inverse problem and find which forces have to be applied. This, making the inverse matrix, it's computationally not stable. So we use some approximation methods here like GMRS. But we will use that because in the project that I'm talking about, we are looking at very short time scale. So we want the cells to be immobile, right? If they are moving or if they are dividing, that will mean you will have to calculate this flow every iteration or when the division happens. So we want to simplify, we assume the cells are immobile. So we want to impose the velocity on the boundary of the cells equal to 0. But then we have to calculate what forces we have to use. So if you use this GMRS, we can end up with this picture here. So in order to have the cells immobile, also the 0 velocity on the boundaries, and then velocity on the left, the capillary side, so we have the proper influx. The red arrows show you what kind of forces we have to apply, and then with those forces, we can calculate the fluid. Okay?
At the top and bottom, what you said, is that a no flux condition? Like is it a periodic condition, a no flux, or?
So we decided to have just the 0 velocity over there. We wanted to have the periodic, but because the cells are cut in different places, the periodic didn't work for us. If you make more symmetric tissue, then the periodic could work. So we just really make like a channel, right? We put the So with that, right, if we put our molecules, they will just go with the fluid. And because there is a zero velocity on the cell boundaries, those molecules will never cross the boundary, right? They will just go with the fluid.
The short question, do your cells move or are they stationary?
No, they are stationary. We want them to have zero velocity.
Well,
Right.
study where we have the slow fast time scales, right? Because you said that the
Right,
flow,
so,
you're doing a very short time, the flow calculation compared to, say, cell time scales.
right, so so if we look at just at the day, for example, right, couple of hours, then we assume the cells are not moving and not dividing. Right, yeah. The nature of the fluid. So this is the interstitial fluid that comes from the vessels, and the vessels are leaky because they are in the tumor. So that's the plasma is leaking from the vessels into the tissue, also because there is a pressure difference, right? What's in the vessel and what's in the tissue. And then, yeah. Well, yeah, but we model this as Newtonian, right? Yeah. I mean, they are simplification, that's true. So then on top of that, we can add the diffusion, which is modeled as the random walk with the diffusivity coefficient D. But here, when they diffuse, we have to take into account that they don't cross the boundary accidentally, right? So if they do, we are just keeping them when they were before they cross, if we don't want the uptake. Okay, so with this model, we are then interested, does the tissue architecture actually plays a role in this transport or not? So I'm showing you three pictures of three different tissues. There is a sarcoma and glioblastoma and pancreatic cancer. The two on the middle and the right, all this blue stuff is the collagen. It's a marker for collagen. On the left-hand side, you see the nuclei and some of the fibers of the ECM. So those tumor tissues look differently. So we created a set of our tissues. So we decided to look at the different porosity. So each tissue in the row has the same porosity value, and this is the area of the void space in between the cells. And then each tissue in the column has the same cellular density. And here, those configurations are regular, so this really means the number of cells in the column. Right, so you could see visually that maybe tissues, when they have a lot of small cells and a couple of large cells and have the same porosity. So with that, we decided to test different properties. So we have one regular and four perturbed tissues when we just tweak the cells locations a little. We use 6 porosity values from 40 to 90%, 5 densities from 2 to 6 cells per column. The velocity of the fluid was fixed, and then we consider different diffusions, and those correspond biologically to like small molecules and then antibodies. This is one simulations that you could see of how those biomarkers, you cannot see much. So how they are moving. So the model actually is what's on the top, right? Of those red dots, that's on the model. But then when we compare this to the fluorescent biomarkers, we found it easier to talk to collaborators when we have this spread here. So if we change the number of molecules into the densities. So this cube here is our parameter space, and I will show you like roughly the cross section of that. So in this picture again, there's the same tissue in every row, and we are changing the diffusivity in every column, and the velocity, imposed the velocity is the same. So what you could see that the tissues on the left are really diffusion dominated. The first column, for example, they diffuse so fast that at this time, most of them actually left the domain. But in the other columns, you could see they are diffusion dominated. The one on the right are advection dominated. The molecules only follow those corridors of the fluid. But the one here in the middle, there is kind of like a jump. There are some that look more like the diffusion dominated, but there are some here below this blue line that show more of those fingers or corridors. And we put here the speckle number, which is the ratio of the advection into diffusion. So for the small speckle numbers, we have diffusion dominated. For large, we have advection dominated. And there is a region that it actually depends on tissue architecture, right? And as I said, they correspond to different size of drug molecules. So you could calculate the diffusivity, for example, knowing the weight of the molecule. There are some ways to measure the pressure of the interstitial fluid in the tissue, but what we try to argue is you actually should know a little more about the structure of the tissue to know. So this is 2D. Yeah, this is 2D. Yeah, that's true. So one big difference, for example, would be if you have molecule that it's stuck here, right? In 3D, it may have way to go up and down, but in our model, it will be just stuck near the cell when there is a zero velocity. Okay, so yeah. So with this model, we wanted to look at this biological problem of the fluorescence biomarkers. So our collaborators are developing the ligands, which are conjugated with the fluorescent dye. So they have to first find out what are the specific receptors for the given tumor or tumor cell line, then create this ligand, receptor-specific ligand. And they are doing that specifically for this fluorescent guided surgery. So when the surgeon is cutting out the tumor, they see the bulk of the tumor cells, but if they start migrating, they usually they couldn't see it. If they have the fluorescent dye attached, then under the fluorescent light, like here in this picture, they could actually see when those cells are moving and could maybe cut them. And then the collaborators use different systems like this.
What kind of surgery is it on the picture?
This is mice. So the pancreatic cancer in the mice in the pancreas, they added the conjugated fluorescent dye ligand that they developed, and then the surgeon, the biologist, was cutting the tumor.
It's the same mice, yeah.
It's what?
The board. So it's like a tumor of the same mice. Right?
Yes, there's a tumor in the mice. So when they are developing those biomarkers, they have to have two cells, one that express the receptor and one doesn't. And then they grow those two cell lines in the mice and then inject the biomarker. And then for those cells that express the receptor, they want to look farther away from the vasculature. So at the beginning, there is no fluorescence, and then with time, the cells actually uptake the biomarker. And for the cells that don't have the receptors, they want to look near the vessel, so they see the biomarker in the vessels, but then with time, there is no uptake, right? Because the cells have no receptors. And you have the fluorescent quantification of those two processes here. So from the high concentration near the vessels for this untarget tumor, or from zero concentration to the high concentration when they look on the targeted cell lines. So we did the similar thing with the model. So we put the window, if there is no uptake, we put the window near the vessel and then reported what concentration do we see of those biomarkers when the cell uptake. And we have very simple model for uptake. The biomarkers have to be close to the vessel, to the cell boundary to be taken up. Then with this window, we have this other trend. So they look similar. They are normalized, but completely different numbers, right? So this is only qualitative comparison. Okay, so this is how we were using the fluid structure interactions method to the fluid structure of our microenvironment. So let me now switch the gears and tell you a little. Yeah,
Is it like typical CMT? Like, is it like for most cancers or for a large group of cancers?
the
Or is it
fluid
like,
part or
specific for,
the fluid part
so this one?
or this the
Oh, yeah, yeah. So it's like more or less universal or?
so sorry, you are taking the fluid part or the
CME.
the tumor microenvironment is typical for every cancer. Yeah, every cancer. The
It's typical for it.
yeah. The fluid, maybe it's more like in the brain, for example.
Oh.
There are some models that use fluid dynamics in the brain. In the other, it's not so popular to use. Most people using diffusion. But if the biomarkers are large molecules or the drugs are large molecules, they may be affected by the fluid part. The small one will mostly be the diffusion. Okay, so now I want to talk about extracellular matrix. So the extracellular matrix is like a structured substance that fills the space in between the cells. It could become, I mean, there are known about 300 different proteins that could form the extracellular matrix. We will be focusing on collagen because that's the one that could be visualized with the second harmonic generation microscopy. So what you could see here in color or in black and white are the fibers of collagen that are visualized with this method. The ECM provides the support for all the cells, but also can trigger signaling. And this organization is affected by 4 processes. So there is deposition, a lot of that by fibroblasts. There could be molecular changes. I will show you in the moment the different tumor-associated collagen signatures. There is a degradation when the cell, some of the cells, for example, can degrade the ECM with the MMPs, and then the physical remodeling that I show you a little more with the model. So what we was become interested in are those different patterns of the ECM around the tumor. And then the Patricia Keeley group identified or named those 3 different patterns. So they are all called the tumor-associated collagen signatures or tags. Tax 1 is the wavy, maybe I show also this. It's like the wavy structure that is loose and not stretched, and it's far from the tumor. Tag 2 are the stretched fibers, usually align to the boundary of the tumor. You could see tags 2 here, here. And then tags 3 is the pattern that is radially from the tumor, and it's often used for cell migration. So you could see some of those patterns here as well. So we wanted to model that, to recreate those collagen, those tags, but also to see in the image you have just the one picture. So we want to see if we can hypothesize how they can change. So this work was done by the undergraduate intern in my group, Sharon Punja, and Anna Forera Pinto, who is the graduate student in my group. And this is in collaboration with Mehdi Damaghi from Stony Brook. So let me tell you a little about the model that we are using. So this is the agent-based model. You saw the beautiful presentation by Paul. So there will be some overlap, I'm sure. So our cells, when they relocate, it depends on the forces that are applied to the cells, and then depends also on the viscosity, inversely proportional to the viscosity of the medium in which those cells are sitting. The F forces are the cell-cell interaction forces. So if the cell divides, they overlap. We want the repulsive forces for them to push away so they don't overlap. You could see some of those blue forces here. And then we have some small adhesion force because we want to form the cluster and don't want the cells to grow in the, when they divide in the random direction. So we want them to form more compact cluster. The G forces are the cell ECM fibril contact guidance forces. So when the cells are moving, they see different orientation of the fibers, and they could, or we could decide, depending how we define the parameters, whether the cells will follow the ECM orientation, or if they will have some persistent migration direction and they will just go, the way that we assign them, or biology assign them. So that depends on this alpha parameter. So on the other hand, first maybe I show you this. This is really bright, so you cannot see very well the first one. So those are the 4 still images. I show you the movie in the moment when we change the alpha, and then this one, which is not very well visible, the cell is just following the fiber directions when the alpha is equal to 0. When alpha is equal to 1, they just go straight to the left top corner because that's the persistent direction that we defined, and then the others are in between. So I show you the movie. There will be 3 cells. It's unfortunately the first example, so let's see how this will be visible. So they are moving, alpha is equal to 0, so they are moving, they're just following the direction of the fibers, not the left top corner that they were prescribed. Especially this one on the bottom is funny because it just goes in circles. So, but you also see, I am leaving some trace on the extracellular matrix, so we have also equations for the ECM. So this is modeled as the vector field, and again, the bright is not so visible. As a vector unit vector field, and then we prescribe then another scalar, which will be the stiffness of the ECM bundles, and then could undergo remodeling. If the cells are growing, they will push on the ECM, and again, unfortunately, you cannot see on this image, but there are fibers that are aligned perpendicularly to the cells. When the cells are moving, they will pull on the fibers, and then there will be ECM that is aligned in the direction of the moving cells. And again, we can change this beta parameter, which is the compliance, and then the ECM can be remodeled more or less depending on this value. And we also can change the stiffness when the cells are pulling or when the cells are pushing on the ECM. So with this, we would like to first recreate those 3 different tags. So the first one, the tax one, what we did is we defined the random direction of the fibers. They are all uniform and have uniform small stiffness. For the tax 2, when the cells are growing and pushing, in the histology, you see those ECM fibers aligned, like encapsulating the tumor, and that's what we are trying to recreate as well. And then for tax 3, when the cells are pulling, they change the direction. You could see here those are more stiff, so they are darker. They change the direction and the stiffness of those fibers in the direction when they are moving. So as I said, we recreated those 3, but in the biology, you don't see the transition because if you have histology, it's, take it from sacrifice the animal, or you have the section of tumor, human tumor, patient tumor, and you don't see the progression. With the model, we could hypothesize what should be done, change to see those progression. So our hypothesis is that the cells respond to different stiffness of the ECM. So we have the cell, and if the ECM stiffness is below the division threshold, the cell will divide and then will push on the ECM, right? And can remodel, can change the stiffness. Once, they change the stiffness, if the cell is above the division threshold and above the motility threshold, that it will migrate and start pulling on the ECM. So we hypothesize that maybe like 2 values of the ECM stiffness that will regulate the cell behavior. So the first one, the division threshold, it's known in the biology that the tumor could be encapsulated, and then the cells could stop dividing. And I show you the image from Valerie Weaver lab when the stiffness was high, the cells actually, instead of growing, they start migrating. So we have some backup in biology with that. So this will be the simulation that will show those transitions. So the cells are growing, and then first they are pushing. the ECM stiffness increases, and at some point, the stiffness is so high that the cells start moving. But some of them, in some places, that is not high enough, so you could still see the cell growth. And the persistent direction here is just out of the center of the cells, right? So they are all moving outside of the center.
So when they migrate, they have to push their steps, the stationary cell.
So when they migrate, they pull on the ECM and they move away. If there will be a cell behind that will like bump into the cell, yeah, that it may push it. Well, there will be there may not, because there will be the repulsive forces between the cells.
Cell is stiff, and
So this one I don't know, but the cell can migrate. They could change the shape, you know. Yeah, my cells are just the circles, but the cell can change the shape. They have lamellipodia that they pull on those ECM fibers, so they can do that. So since we have a model, we can run different parameters. And this is something that I called from my previous research the morpho chart. So we change the parameters, you look at the morphology on the result, and then we could classify. So if the growth arrest stiffness is very small, nothing is growing. If the migration stiffness is very small, everything is migrating. When the migration stiffness is high, then you have the cluster of cells growing, and there is no migration, and then there is this region in between. So some of those morphologies are also observed in the literature. So I am showing you 3 images from the published paper, not from our collaborators, that you could see that. There may be a question why all of them are moving to the left and not to the top. So there are 2 ways to deal with this kind of models, right? Either you run a lot of simulations and you try to average in order to compare. For us to compare, we fix the random seed generation at the beginning and then just vary the values, right? So we didn't want to have too much variability if those random numbers that decide where the cells are placed when they are dividing, for example. So I think that's the reason that we have the same funny shape, that they only migrate to the lower left. Okay, so this is how we model the ECM structure. So, and this was published last year, and the other paper was published many years ago. So now I wanted to tell you something that is our ongoing work. So this is unpublished work, and I want to, so in those previous models, I was focusing on fluid or focusing on ECM, but of course, there are other components of microenvironment then too, so you cannot just separate one and do the model. So in this one, we will have the 3 remaining components. So I will show you how we are using the histology of mice bladder to build the model. We will be focusing on the lymphocytes here as a cellular structure and on the oxygenation of the tissue as our chemical structure. So this is the work that we done in collaboration with Shari Pilon-Thomas and Michael Pogg from Moffitt. It's the project on the adoptive cell therapy. And we've heard about the adaptive cell therapy, when you adapt the treatment to the tumor behavior. This is adoptive cell therapy, when you adopt the T cells from the patient and then inject them back into the patient. And this work was done by my postdoc, Morik Ojwang, and we have the high school student. We are running the summer high school HIP-IMO program in the summer. So Jay took the Morik model and then do some of the cell migrations, which I will show you. So this adoptive cell therapy, the tumor is resected from the patient. Then our collaborators divide this into small parts and want to grow the T cells from the tumor. So those T cells in the tumor are there because they were attracted by the tumor. So there is a hope that they are really reactive to the tumors and they will be good killers of the tumors, but not all of them are. So when they expand those T cells, they have to then select, so expose them to the tumor, expose them to the tumor factor specific for the given tumor and see which of them will be reactive, which no. Those that are reactive are then grown into the large numbers, and then it can be injected back into the patient. So the patient will get their own T cells just in the larger numbers, and sometimes they expose them to some other factors to make them more functional. Now, in general, when you do that, you have to deplete the patient of their own T cells before you inject those TILs. So this is the lymphodepletion with the radiation therapy. In the project that we are part of, we are looking at the bladder tumor. So actually everything can be done locally through the catheter. So we don't need to use this lymphodepletion. And if you do the lymphodepletion, you can only do that one. This is really harmful for the patient. So in the bladder, we do not need to do this lymphodepletion. And there is a possibility that we can put multiple injections of the TILs and the possibility of do some combinations because everything will be done locally through the catheter. And as a part of this project that we are collaborating with, we actually got the funds for the Dr. Pop got the funds for the feasibility clinical trial that is now at Moffitt. So we will have 12 patients over 4 years, and then, well, it says 2 years here, but it's over 4 years. And then the goal is to see whether the injecting the T cells through the catheter to the patient's bladder, the bladder cancer patients, will be safe. But we could use some of this data to our modeling. I will not be showing this here. So as a part of this preclinical, so we have the project in the preclinical in mice and then this clinical trial with patients. So I will be talking only about the preclinical model. So this is known that if the T cells are in the harsh microenvironment like hypoxic or acidic, they have lower viability, they are less proliferative, they are less functional. The in vitro experiments actually show that they respond to the migration cues the same way, but all the other functions are diminished. So the graduate student in Sharipion Thomas lab did the experiments when the mice through the catheter the same way like that could be done in the clinic were exposed to combination therapy, the gemcitabine and then the OT1 cells, which are the cells that recognize the bladder cancer in the mice. And of course, they do the untreated as a control and then did the monotherapies as well. So from these studies, we see that the gemcitabine itself didn't help much. The adoptive cell therapy itself helped a little, but not too much. And then combination was much better, but still it's like plateau, right? It didn't kill the tumor. So we were interested why it happens, why the OT1s are not much better than gemcitabine or the untreated. So our interest was to see, okay, where those T cells are actually in those tissue, right? We see in the size of the tumor, there's not much difference. Is there any difference in the tissue? And to see why they are not effective. And the long-term goal is that we want to use our models to actually test different scheduling, different maybe combination and see if there is something that will help. But that's preliminary data. We don't have the answer yet. So in order to build this model, this is our pipeline. So we have experiments from Shali Piron, Thomas lab. We can take the tumors are resected at the end of the experiments. We can slices of those tumors. We can stain them. So we are interested in where the cells are located, so H&E, where the vessels are located with CD31, and where the T cells are located with CD8 antibodies or staining. Now, those tissues are then segmented. We got all the coordinate sizes of the cells, and then our goal is to calculate this oxygenation of this tissue. So here is example, again, have to change the color scheme, but the H&E is all the pink, so cannot change that. But you could see there is a shape of the whole bladder, and I show you in the moment where is the tumor here. But it doesn't fill the whole domain, right? So we have to do some pre-processing. So we have to identify those points outside of the tissue, so we don't calculate diffusion there. The bladder has actually cavities, so you have to identify those areas inside the tissue to don't calculate the oxygenation there. And then we use the reaction diffusion equation. Oh, I think I didn't mention in this graphic which part we are using from our micropharmacology model. So we have tissue, we have diffusion, and we will have just the membrane uptake. So our diffusion in this space, the oxygen distribution in this space depends on the diffusion, and then we model the cellular uptake with the Michaelis-Menten method. We just, because we are like off lattice, right, we just have to make sure that we only uptake where the cells are located or within the cell area. That's what those indicators functions come from. Then for the vessels, we treat them as the boundary conditions. So we will input the oxygen level in those points, the same level throughout the simulation. So we don't model the flow, changes in the flow in the vessels.
Where would the cells, like on the boundary, pink blue here in the graph?
The tumor cells?
For, the boundary, that's what you refer to boundary cells.
So we have diffusion in the I know in the domain, right? Except of those pink and cyan points. And then we have a lot of cells. I will show you in the moment another picture when the cell will be uptaking the oxygen, right? But then we have cell with the center and with a radius. So we have to make sure that in all the points in the domain that are inside the cells, the oxygen is absorbed. So that's what we use those indicators functions. So right, we say, okay, if the point on the domain is within the radius of the cell, then we will use the Michaelis-Menten equation. If the point is far away from any cell, then it will be zero. So it's not the cells on the boundary.
Characteristic sets.
But for the vessels, we treat them as the boundary conditions. So
To see on the diagram, where are they? Are they interspersed within the cellular points and it's just hard to tell the difference, or?
the vessels?
Yeah.
Yeah, so all these red points are the vessels. And you don't really see here the cells.
Okay.
Maybe in the next picture,
Okay.
let me see. It's kind of hard
Okay,
to show everything. Yeah. Let me see. So I have the next picture, which has more of those individual elements. But now our question is, okay, so we would like to know what's the distribution of oxygen in this tissue, right? We just have like an image. We know where the cells and the vessels are. So where are the hypoxic areas? Where are the well-oxygenated areas? So what we decided to do is to run, so you have influx of oxygen from the vessels, there is diffusion in the tissue, and then the cells are absorbing the oxygen. So we decided to run the simulation until the L2 norm between the two consecutive landscapes is very small, right? We choose 10 to the minus 10. So we start with this configuration. You have hypoxia in those outer and inner points. You have zero in the tissue, and all these red spots are the vessels. And then we run the diffusion, and we calculate the L2 norm between two consecutive iterations, and we want this difference, which we call error, but it's really difference, to be less than 10 to the minus 10. So they are numerically stable, right? There is not much difference between the two consecutive iterations. And that will be our map of oxygenation. So we did some sensitivity analysis. So the one question was, okay, we put 0 concentration in the whole tissue, does it matter? If we put 0 or 60 is our value in the vessels or something in between. So actually it doesn't matter. They all converge to the same value. Some of them converge faster, some converge slower, but they converge to the same value. Also, it's not, if there are small perturbation in the Macaulay-Menten parameters, there is also not much difference in the outcome. If the maximal uptake, it's largely perturbed, that you see the difference. Of course, they are sensitive to the value of the influx from the vessels, right? If you put 0, then there will be no oxygen at all. So this is sensitive. And they are sensitive if we change drastically what's in the outer or inner cavities. So we decided to keep this hypoxic. We know that the urine in the bladder is acidic, so we assume there could be also hypoxic. That's our assumption. So a question about the cells, right? So the cells are really dense, so it's hard to see. So this is the histology image. The yellow part is the tumor. So all of those are the tumor cells. They are really dense. The lighter pink are the normal cells that we also use in our simulation. You could see this oxygenation map. And then we were interested, where are those T cells, right, in this oxygenation map? So all those blue points here are the T cells. And here in this picture, you could see T cells that are in oxygenated areas in red, and T cells which are in the hypoxic area in blue. And this grayish part are the hypoxic tumor cells. So the tumor cells are everywhere. Maurik was just drawing the hypoxic cells for, so we can compare where are the T cells. And then some of the statistics. So we have about, maybe 20% of T cells in the hypoxic areas, and majority is in the normoxic or physioxic areas. And the ones that are close to the vessel are in more oxygenated areas, than the ones which are farther from the vessel are in more hypoxic areas. So this was the case of OT1 monotherapy, right? So the tumors were grown, they measure them at day 7, then the OT1 were injected at day 14, and then we wanted to see early after injection, so at day 17, those are the data that we are showing. We also look at the other treatments. So we have untreated case on the left, and then just gemcitabine monotherapy on the right. So of course the untreated are larger, they are more hypoxic, but if you look at the T cells, again, the majority, there's more in the hypoxic, well, 25% still, but majority is still in the well-oxygenated areas. The one with gemcitabine, there may be less hypoxia comparing to the untreated, but if you look at the T cells, they are still in the well-oxygenated areas. Right, so
That's okay.
it's not published yet, so it's prepared to publication. It's on my desk. So we look at all the data that we could get from the biologist, right? So the first experiment they actually ran for 25 days. And then we look at the data, and there was not too many T cells. So then we ask, can we look at the earlier points? So that's what we get the data at day 17, so like 3 days after injection of the T cells. And in all of them, when you look normal tissue, we look at the normal tissue, monotherapy, combined therapy, untreated. The normoxic one are all in red, right? So they are like close, 75, 100%. The one there, some they are in the hypoxic areas, but they are really the minority. So those T cells just don't like the hypoxic areas, right? No matter what how you treat them. And to be honest, if you think about that, if there is only gemcitabine monotherapy, there is no injected T cells. So the T cells which are there, they're native T cells, and they still don't like the hypoxia, right? They only sit in the well-oxygenated areas. So why the combination was better? If you look at the combination, right, it's still majority is in the oxygenated areas, whether at the early time points or the late time points. So we look at this tissue, and yes, there are no cells in the hypoxic area, but there are no hypoxic area either, right? All the tumor is much smaller after the combination, and this is all well oxygenated. So that's what we started with, the analysis of the data that we have. And then, so I show you this simulation. This is very preliminary. It was not calibrated, but it was done this summer by our high school student. So we took the same tissue, we take the part. So, you know, they're high students, we cannot ask them to run simulations for a couple of days. So we make the smaller domain, just look at the subset which has the cavity. And Jay actually created the acidity map in the similar way like Mori created the oxygenation map. And then he put 4 or 5 rounds of the T cells and created the simulations how they enter the tumor and start killing the tumor. And the green one, the active one, the red one, the exhausted one. And he used the different pH levels to make the T cells less active in the high acidity. So this is something that we are going with, right? We want to use this kind of models to actually see if we can improve of the treatment. This, again, it's uncalibrated. Okay, so I show you the 3 examples. We use this kind of modeling in other, so if you are interested, I'm happy to talk after that. So we looked recently at the hypoxia-activated products. We published a series of papers about the acquired drug resistance with Judith, when we actually identified all of this is more like in the in vivo setting. So we identified areas in this in vivo setting that this acquired resistance can arise. We look at the IC50 curves in 2D and 3D experiments. So this will be in vitro. And we also look at the other targeted biomarkers. So if any of those is of interest, I'm happy to talk after. So I told you about the tumor microenvironment. I show you the picture of what we think are the elements. The color one was made for the presentation. This is the original picture in our review. And I'm happy that we are not the only one who think like that about the microenvironment, because there was the recent review from the Rakesh Jain group. And Rakesh Jain is like the father of the bioengineering in cancer. So they published, his group published the paper when they didn't talk about the hallmarks. They called this physical traits of cancer, but they identified the fluid pressure, the solid stress, the stiffness of the ECM, and the microarchitecture as the important factors for the tumor development and tumor treatment. So for example, if the blood vessels are leaky, the fluid pressure will increase, if the cell proliferates and actually produce stress, and then the vessels could collapse. When the matrix is deposited by fibroblasts, for example, the stiffness of ECM and the microarchitecture can change. They also connected all these 4 traits to all the different hallmarks of cancer. So I will not talk about that. If you are interested, they have a lot of examples of how they influence one of the other with the hallmarks of cancer. So what I would argue is that these hallmarks of cancer that were published are really hallmarks of cancer cell. And then maybe we need additional hallmarks of cancer ecosystem. Well, the set of hallmarks then is getting larger and larger. So and another argument may be that maybe you want to reduce them and not increase the number of hallmarks indefinitely. So, but we definitively, the cancer ecosystem was the phrase that was here before I came to talk. You guys were talking about that, so we think about that like that. Okay, so I think I named the people who are doing the work when I was presenting. And when I take questions, I just wanted to leave you with some advertisement. If you are interested what we are doing, we have a lot of different activities. If you have undergraduate students that are interested to come and work with us during the summer, we have the Spark program, we have the graduate program. If you have undergraduates that want to do the interdisciplinary work, Moffitt is organizing once a year the MIOT, Moffitt Innovators of Tomorrow, so you could apply and come and visit and talk to the PIs and see if you like. And then we are hiring postdoc. I am hiring, and other people in IMO are. So if you are interested, just talk to me. And we also have the faculty position. I think it's not open yet, but we'll be hiring another faculty. So just talk to me. Thank you.